Thanks a lot. So we're going to start with the session on structural bioinformatics, or continue on the section on structural bioinformatics and biomolecular simulations. And now if we get the next slide, I'm going to introduce Pilar Cossio, which is going to be the keynote speaker for this session. Pilar studied physics at the University of Antioquia, Medellin, Colombia, and she finished in 2007. She then traveled to the CISA institution in Trieste, Italy, where she did a PhD in computational biophysics under the supervision of Alessandro Laio. In 2012, she began her postdoctoral degree at the NIH USA in experimental and computational biophysics under the supervision of Dr. Gerhard Hammer. And in 2013, she continued the postdoc at the Max Planck Institute for Biophysics in Germany, also under the supervision of Gerhard Hammer. And since 2016, she leads her own Max Planck tandem group associated with the University of Antioquia. Currently, she's directing a postdoc, a PhD student, and 2 master's students. And we're very happy to have you, Pilar, and to have you share your work with us. Please go ahead.
Okay, so thank you very much for the organizers. I'm really happy to be here. Muchas gracias a todos por la invitación, as all the speakers. Doy mis gracias en español. En realidad es un honor estar aquí entre tantas mujeres y tan talentosas. Entonces, muchas gracias. Thank you very much. So today I'm going to talk to you about in silico drug discovery and peptide design. And this is basically a work that I started in my last 4 years when I became a Max Planck tandem group leader between the Max Plancks in Germany and the University of Antioquia here in Medellin, Colombia. So I'm going to start with the motivation of my work, what motivated us to start studying and doing in silico drug discovery and peptide design. Then I'm going to present the theory and results, basically the methods that we've been developing for tackling these problems and addressing these lines of research. And finally, I'm going to present the conclusions of our work. So when I started the group leader position, they told me, okay, you have to work on tropical diseases. I hadn't really worked on drug discovery or peptide design before. I was coming from the side of molecular dynamic simulations, protein folding, also cryo-electron microscopy, so very different lines of research. But these numbers shocked me when I started, and it's that tropical diseases affect more than 1 billion people, and there are over 170 deaths per year, and there's more than 100 countries that are directly affected. However, most of the countries are from tropical areas, from countries that are in developmental phases or that are very poor. So there are insufficient funds to study and develop cure for most tropical diseases. And even more now in the times of COVID, tropical diseases and neglected tropical diseases have received much less funding than the years before. So basically, there are still many diseases that affect more than 1 billion people, but unfortunately, there's not a lot of funds. So the motivation and the goal of our work is basically to provide and develop computational methods that can complement or replace some expensive experimental techniques for drug and vaccine discovery. So I think nowadays, computational methods, as we've seen in this lecture, as we've seen in this workshop, are at the stage where we can provide new insights and help discover new drugs, help in the development of synthetic vaccines to replace or complement some expensive experimental techniques. We heard before in the lectures that developing a drug takes 20 years, costs billions of dollars. So how can we help from a computational perspective in this very challenging task? So our research, I just want to tell you a little bit about our research lines. So we have some lines of basic science, and basically we do molecular dynamic simulations, structural techniques, structural biology techniques with cryo-electron microscopy, and also some methods for understanding single molecule experiments. And we try to gather all of the things that we've learned so far with, like, say, this basic science, and apply it for drug screening and peptide design. And so now what I want to talk to you is about these two lines that we've been developing. So, so now I want to show you the theory and the results. So this is the drug screening line, and this is work done by our postdoc Isaías Lanz and a master's student that is now in Paris, Caris Palacio. And the idea is to filter molecules that can bind to a protein target. So how, from a huge database of molecules, how can I find proteins that can bind with high affinity to a protein target? So this is like the main goal or the main strategy of our work. And so we have a funnel-like procedure in which we start with a large database of structures of molecules, so a large library of molecules, of potential molecules. And then what we start doing is we start applying filtering layers. So say that we start with 100% of molecules here, then we hope to filter the best 50% of molecules, then here in this stage the best 5%, and then obtain a very small set of molecules that we can give to our experimental collaborators, and then they can test and really validate. But basically this funnel-like strategy avoids having to test say hundreds of thousands or millions of molecules in the experimental lab, and just reduces the set to a small set that we have we consider have a much higher success rate. So our first stage consists of a pharmacophore-based virtual screening, which in the first lecture, we heard a lot about pharmacophores. Then we're going to do molecular docking and consensus scoring in the second filtering stage. And in the third filtering stage, we do molecular dynamics. So what I'm going to try to explain is each of these stages separately, and for you to take the key ideas home. So basically we have a developmental phase where we optimize and basically developed all the methods to do this filtering and molecule filtering stages, and then an application on the FAD synthase. So the challenges are, how do we sample the molecules? How do we select the best molecules? How do we also sample their conformations bound to the target? And also, how do we predict in the end the affinity of these molecules to the target? So in the first stage, we developed a method called Flexi-pharma screening. And so the idea is to build a set, say in the back you see the protein target. And here what we're going to do is use AutoGrid to create a set of pharmacophores representing different types of atoms. And these pharmacophores are an ensemble of physical chemical features that should represent what the molecule that binds to this structure the important features that it should have. So then we take the pharmacophores that we generate, and we use the program Farmer to screen very quickly through a library of compounds. Okay, one of the problems here with pharmacophore screening is that basically this program Farmer tells you a binary output, which means this molecule matches or does not match the pharmacophore, but this doesn't let you rank or classify the molecules if you want to take, say, different percentages of the library. So in our Flexi-pharma method, what we do is we take into account the conformational ensemble of the receptor. So for example, we take conformations from molecular dynamics simulations, then we generate many different pharmacophores from each conformation, and then we see if the molecule matches or not at least one pharmacophore from each of these conformations. And then we apply a ranking by vote strategy. So the molecules, and basically we're ranking with respect to the conformation. So the molecules that match more conformations will receive a vote. And so we use these votes to basically classify the molecules and rank them according to the number of conformations that they match. So, so this is used to rank and filter the molecules. We tested this method over 20 different systems with ligands, like known ligands and decoys. And we find that for 19 of them, we have an enrichment of the database, which means that in this subset of say I take 5% of the top ranking molecules, I have many more ligands than decoys. And so this is very encouraging for our first step, that is a very, very fast computational screening. So in our second stage, we use molecular docking. And what we have here in molecular docking is not only like pharmacophores, but really now one tries to fit the molecule inside the receptor and score the molecule. However, what we found is that basically, if you look, for example, you compare the rank or the score of autolog BINA with respect to, for example, ICM, we find zero correlation. And basically here, the red dots are the ligands and the gray dots are the decoys. So ideally, if docking were perfect, we would have a perfect correlation, but here you can see that there is not a lot of correlation. So we need to apply a consensus ranking strategy. So what we do is say we take, for example, 6 different docking programs, and we try to say, okay, what is the consensus saying? So typical consensus would be just take, for example, the rank or the average score and take the average score over all the programs. But this acts as a conditional and. So you would be taking like the yellow points in this graph. But so what we thought about is, okay, so what if we have a function that acts as a conditional or? What if you have a scoring function that is really, really bad? Then if you use like the standard consensus, that this would completely destroy your ranking and your like classification procedure. So what we did was we developed this method together with Claudio Cavazotto from Argentina, called exponential consensus ranking. And basically what it does is it acts as a conditional or in order to classify the molecules, taking into account all, and not all, but most of the best scores of the docking programs. So we apply this also to many systems, and we find that, let's say, the exponential consensus ranking is at the top, independent of the system or structure, which is in many cases you see that there's biases due to training or things like that. Okay, so now the part of the application. So this is a work in collaboration with Milagros Medina. And we've worked on the FAD synthase. So FAD is a redox cofactor. It's used by enzymes to accept or donate electrons. And in humans, FAD is synthesized by 2 different enzymes, right? So you have basically 2 different enzymes that synthesize FAD. However, in prokaryotic systems, FAD is synthesized by a single enzyme, which is the one you see here. And the interesting part is that this FMNAT module has no human homologs. So it's a really, really interesting target for tackling these pathogen microorganisms like tuberculosis or pneumonia. So basically we're going to apply all of our protocol over this enzyme that has no human homologs for looking for new potential drugs. So this is the funnel-like protocol that we apply. And first, what we do is we have a training set in which experimentally they tested around 2,000 compounds and they found 37 ligands. We try to optimize all of the stages. We have a final stage that does molecular dynamic simulation with each of the compounds and scores. And so we basically optimize everything with this experimental data set. And then we apply it over a new compound library with 14,000 compounds, and we only select 17 for experiments. So all our experimental collaborators were very happy because they didn't have to test 2,000 molecules. And in the end, we find a very high enrichment, like 8 times better than before. And we find that 6 molecules out of 17 present inhibitory activity against this model of the FAD synthesis. So we're really happy because we think definitely there are things to improve, but this is a first milestone in our work. Okay, so now I want to talk to you about this peptide design project. And the goal is to design peptides that bind with high affinity to the major histocompatibility complex class 2. This is a molecule that lies on the cell surface and basically grabs exogenous peptides, displays them, and then basically triggers immune response, T-cell response, immune response. So it's key for synthetic vaccine designs to trigger with higher like potency immune response. So here I also want to talk to you about two phases, about the protocol, what the protocol consists, how we've optimized it, and also about the applications. So the challenges are exactly the same that we faced in drug discovery. So how do you sample this huge space of sequence? Say that you have a peptide that's 10 amino acids long, then you have 20 to the 10 possible combinations. We want to explore as better as we can the peptide sequence, but we also want to explore the binding conformations of these peptides to the MHC class 2, and we want to also predict better the affinity to these molecules. So this is the protocol. So we start with an initial structure of the MHC class 2, which is the orange and blue protein bound to a peptide. And so what we do is we perform a Monte Carlo algorithm in which we choose a random point on this chain. Say we chose this one, this position, and then we perform a random mutation. So say that we mute to, for example, alanine, and then we sample these conformations with molecular dynamic simulations. We score these conformations using also consensus scoring and multiple scoring functions. And then we accept or reject this mutation according to the score. And we repeat this many, many, many times following a Monte Carlo scheme. This is a better example of how the protocol looks like. So basically the red amino acids are the ones that are rejected, the green ones are the ones that are accepted. And the idea is to optimize multiple scores at the same time, optimize this score so we can find, in principle, peptides that have a higher affinity, a higher chance of having a higher affinity to the receptor, to the MHC class 2. Okay, so what we did first is we had to optimize how to do the mutation, how to place the side chain in an optimal way, how to place this rotameter. So we did optimization and benchmark with respect to the MD ensemble, which we wanted to reproduce. We also developed a method to be able to predict what are the best scoring functions and how to combine them in order to be able to rank peptides, different peptide binders according to their IC50, given some training benchmark system. And now this is what we're doing. This is basically still work in progress, and we are trying to do the proof of principle, and it's start with a peptide sequence that we know has a given affinity, apply all our protocol or all this machinery, and be able in the end to actually we already have the experimental data, and we have peptides that are able to bind with higher affinity than the initial reference. We're also working on, like, say, the final goal, and it's to design an epitope or to optimize an epitope to become a universal epitope. So these are 4 different alleles of the MHC class 2. These are basically, you can think about different populations. And so what we've seen is that some peptides bind very well to, say, one allele, but they bind very bad to another. But in order to have like a universal epitope, a universal vaccine that works for all the populations, we want this epitope to bind with high affinity to all of the alleles. So we've been applying our protocol in order to optimize in parallel the sequence in order to increase the affinity to the alleles at the same time. And we're also very optimistic about this work. So the conclusion is, I think, and what we've seen also in this very nice workshop, is that computational methods can really complement and replace some of the expensive experimental techniques, more in and much more needed in developing countries like Colombia, for example, where there's not enough funding to really look for cures for all of these diseases. So we're very optimistic about this. We think that there's much more things that can be improved, but I think we're on a good track. So I would like to thank my group, the people that have developed the work, basically hardworking students. I'm very happy about having this opportunity as a Max Planck Tandem group leader, and I thank you very much for your invitation. Any questions?
Hello.
Hi.
Thank you very much, Pilar, for your talk. That was a great talk. So I would have an opening question for you. When you're showing these methods for optimizing the affinity of the peptides for the binding surface, and you're showing how you perform different mutations and then optimize binding affinity for each, I had the question, how do you decide in which order to do these mutations? Do you explore the whole set of sequence space, because there's so much combinatorial effects between positions in these peptides that they're not so independent? So have you had any strategy to deal with that?
That's an excellent question. And in fact, for the mutation and how to select them, we tried different strategies. So one was doing it, say, completely random, so random position and random amino acid. Another one was focused. So this MHC class 2 has been widely studied with bioinformatic analysis, and basically the core position, you have these matrices that tell you what is the likelihood of having an amino acid in, like, say, position 3 or an amino acid. So we use these matrices as a probability to perform a mutation. So now I'm not going to say all of the amino acid types are equal, but I'm going to follow the probability given bioinformatics matrix. So we did several different strategies. And in fact, this one, the last one that I mentioned, is the one that worked best.
Great. So you're using kind of like the PSSM of the known best binders to guide your search.
Yes.
Great. So we have another question from, I guess, the organizers. I think it's a question from Cristina Marino. She wanted to know, how do you perform these mutations computationally? Do you use a specific program to make the mutation and place the side chain, I guess, is the question.
Yeah, so in fact, like, one of the first papers was basically trying to figure out which was the best of the existing protocols to perform mutations. So we tried, for example, Amber, Squirrel, Rosetta, like all of these types of programs that perform the mutations. And we found that both Rosetta and Squirrel were the best ones to be in correspondence to put basically the side chain where it would be from molecular dynamic simulations.
Great, thanks. So I will correct myself. That was a question from Anna. And I think Cristina has a question. Do you want to unmute yourself, Cristina, and ask your question?
Thank you, Lucia. Thank you, Pilar. Nice talk. My question was, maybe I've lost it when you make the explanation, but did you say that you consider different conformers of the receptors?
Yes, in the drug discovery, yes.
So did you find different responses, and what's your I mean, how do you think we have to do when dealing with several conformers of a receptor?
Yes, so I definitely think, and basically behind all of the drug discovery protocol, there's always been like an assessment of the conformations of the receptor, and we use molecular dynamic simulations. But we found in most of the cases that taking into account the variability of the conformations of the receptor is important. Now, this is something that people have, some people have developed, but it's not very widely used. So I think it's still an open field of what is the best way to take these conformations into account. So here, for example, we proposed a voting strategy. So these are different conformations of the receptor from the molecular dynamics, and we say, okay, if this molecule fits well to many of the different conformations, to many of the pharmacophores created from the different conformations, then we think that this molecule is good. So we give them more votes. But this is still open. Like, how is the best way of dealing with the conformational ensemble is not completely like defined, but I definitely think it's very, very important.
Thank you. Thank you very much.
Thank you.
Thank you very much, Pilar. I think we have one last question from the panelists. Do you want to, whoever it is, unmute yourselves and ask?
No, I think that she has kind of answered, because I was asking Pilar, trying to ask her if how did they select the conformers from the protein. So I was asking you if you selected the maximum pair of these conformations, or which was your strategy, but okay, you kind of answered me.
Yes, so but just a compliment, we selected them every X time steps. So we didn't do any clustering or anything. We just said, okay, every 1,000, like 1 nanosecond, I'm going to take a conformation. So there you hope to like have a good representation of the conformational ensemble without doing anything like clustering or things like that.
Cool, thank you.
Sorry, do we have time for another question?
Yes, we do.
Thank you. So Pilar, you say that X nanoseconds you take a conformation. How
Yes.
long was your molecular dynamics?
Yes, that's a great question. I think that you asked this to the other before. In fact,
I think that to have a representative impression of the structure of a protein, you might need to run many nanoseconds, maybe 500 or 1 microsecond.
Yes, so this was basically, so here we are trying to just sample the conformations very close to the starting structure. So we do maximum, say for the filtering, for using, so say testing 600 molecules, we do only 10 nanoseconds per complex, because basically we can't afford more. If we could do more, but if you have to do molecular dynamic simulations of 600 molecules, that's quite a lot. So basically we limit ourselves to maximum 10 nanoseconds. And also with the design, in the design, we do molecular dynamics and we do between 5 and 10 nanoseconds maximum, because we have, say, 500 different mutations. So that adds up to microseconds, right? But you're doing different complexes. So if we had a big computer, we would do more. However, if you're only looking at like small changes and stability next to the drug or to the peptide, we found that at least 10 nanoseconds, you can reach a stable plateau, things are stable, and then you can do statistics over that. So it depends what you want to really, if you want to look at the minima or if you want to look at the whole conformational ensemble. For the whole conformation ensemble, we don't have time. But around the minima, it gives you, at least it avoids biases that you could have for a starting structure.
Nice, thank you very much.
Thank you.
Thank you very much, Pilar. And there are several congratulations for the talk. It was really excellent. So we thank you again very much for your talk, and thanks for being here.
Thank you very much. Thank you.